everyone, welcome back to our YouTube channel AgriAddict. This is our YouTube lecture series on IBPS AFO mains exam, which is also useful for all the other agricultural exams. I am your mentor Spoorthi, who have completed my BSc Hons Agriculture from University of Agriculture Sciences Bangalore Sub-Campus VC Form Mandya. Currently, I am pursuing my Masters at ICAR IAB Ranchi under the Department of Genetics and Plant Breeding. I have secured All India rank 6th in my ICAR PG exam of 2023 and 2024 in the subject Plant Science. Today, I am here with a new and exciting video on cell ultrastructure, that is part 2 of cell ultrastructure. Before diving into today's video, I like to give a brief introduction of or recap of all the concepts that are covered in the part 1 of cell ultrastructure. In part 1, we saw the definition and importance of cell theory, which is proposed by Schleiden and Schön, and next we studied the major differences that is existing between eukaryotic and prokaryotic cell, animal and plant cell. We also studied about the components of cell, which includes cell wall, cell membrane, cytoplasm, under which the various cytoplasmic inclusions like endoplasmic reticulum, Golgi body, ribosomes, and we also studied about mitochondria. Without any further ado, let us start our today's video with plastids. Plastid is one of the important organelles that is present in the plant cell. They are the discoid-like structure which are about 5 micron in diameter and 3 micron in thickness. These are present only in the plant cells and they are absent in the animal cells. These are present in plant cells because the major function of the plastid is the photosynthesis, which is a vital metabolic activity that is observed in plant cells. And the word plastid is derived from a Greek word plastikas. And the term plastid was first used by the scientist Schimper in 1885 itself. And based upon the type of pigments present and also the function, these plastids are classified into 3 different types. One, it is chromoplast, leucoplast, and the third important type of plastid, that is chloroplast. First, we'll see what are these chromoplasts. Chromoplasts are the colored pigment containing bodies where they contain all the other colors except the green color, that is chlorophyll. They contain other pigments except the chlorophyll, and based upon the dominant pigment that is present, again they are classified into 3 types. One, it is rhodoplast, which is red in color, phylloplast, that is yellow, and xanthoplast, which is brown in color. Try to remember the respective colors with the type of plast. And next, coming to leucoplast. Leucoplast are the colorless plastids, that is, they do not have any pigments, but their major function is the storage. Based upon the type of biomolecule they are storing, again they are also classified into 3 types, that is amyloplast, which stores carbohydrates, eleuroplast, which stores protein, elaioplast, which stores fats or lipids. Next, coming to the third important type of plastid, that is the chloroplast. The term chloroplast is derived from a Greek word which is chloros, which means green, plast means entity or body. As the name indicates, these contains pigment, that is chlorophyll, which imparts green color to the plant, and the major function of the chloroplast is the photosynthesis. It was first observed by Anton van Leeuwenhoek in 1679 itself. And these chloroplasts are found in palisade and spongy tissues where the photosynthesis takes place, and these palisade and spongy tissues are observed in the leaf tissues. There are about 20 to 40 chloroplasts per cell, and these chloroplasts are also a double membrane bound structure where the inner membrane encloses an empty space which is called as stroma. See, here you can see the overall structure of the chloroplast, where this is outer membrane, this is inner membrane. Inside this inner membrane, an empty space is called as stroma. It is the place where actually the dark reaction of the photosynthesis takes place. Next, inside the stroma, there is a presence of structure which is called as granum, where each grana contains a coin-like structure which is called as thylakoid. These thylakoids will be stacked upon each other in order to produce the grana. And thylakoids are the actual site where the light reaction takes place. The chemical composition of the chloroplast is that majorly it is containing 50 to 59% of proteins, 12 to 34% of lipids and 5 to 8% of chlorophyll. Along with that, it also contains 0.7 to 1.1% of carotenoids, 1 to 7.5% of RNA, and remember it contains 0.2 to 1% of DNA. Like mitochondria, even chloroplast is also semi-autonomous in nature. That is, they have their own DNA as well as the ribosomes to synthesize their proteins. And I would like to encourage to say that what the chloroplasts are commonly called. Like as mitochondria are called as powerhouse of the cell, there is also a common name for chloroplast. If you guys know, please post it in comment box. Then now let us see about the functions of chloroplast. The major and the important function of the chloroplast is the photosynthesis, since it contains the pigment chlorophyll, and it is also involved in photorespiration because of the presence of rubisco. It is involved in protein synthesis. As I mentioned earlier, it contains ribosome which is 70S type, which is similar to prokaryotes, and it is involved in electron transport chain during ATP synthesis. And remember that chloroplast is a site where purines, pyrimidines and most of the amino acids are synthesized. Okay, purines and pyrimidines are the actual site where the are the major nitrogen bases which are synthesized in the chloroplast, whereas in case of animals, these synthesis will take place in the cytoplasm. Next, coming to the most important organelle of any cell, that is nucleus. This nucleus is called as master of the cell, because all the metabolic activities that is taking place in the cell will be controlled by nucleus. And it was first discovered by Robert Brown in 1831, and it is a spherical shaped body where it occupies about 2/3 of the total space in the cell. And the size of the nuclei will also vary from one tissue to another tissue. Whichever the cells which is metabolically very active, the size of the nuclei will also be very large. And usually one nucleus will be present per cell, but there are certain tissues where more than one nuclei is also present. But it has to be remembered that in the cells of RBC and companion cells of the phloem, there is no nucleus that is present. Nucleus is the storehouse of almost all the genetic information that is required for the functioning of the cell or the organism. That is in case of eukaryotes. Whereas in case of prokaryotes, the genetic material that is in the circular form will be present in the cytoplasm, which is called as nucleoid. There is no nucleus or nuclear membrane which is enclosing it. And in nucleus, ribosomes, tRNA and mRNA will be synthesized. And there are 3 different types of structures that is present in the nucleus, that is nuclear membrane or envelope, nucleoli and also the chromatin fibers. These are the 3 important structures that is present in any nucleus. First, we'll study the nuclear membrane or the nuclear envelope. Nucleus will be enclosed by 2 concentric lipoproteinaceous membrane. That is, if you consider this as nucleus, it is having 2 membrane, that is, it is a double membrane bound structure, and these membranes are made up of lipids as well as the protein, which is about 70 to 80 angstrom thick. And the space between 2 membranes is called as perinuclear space. This outer membrane, inner membrane and the perinuclear space together, it is called as nuclear envelope. And these nuclear envelope or the nuclear membrane will be connected to endoplasmic reticulum. See, here you can see this is the nuclear envelope where it is a double membrane bound structure, and this nuclear envelope will be connected to endoplasmic reticulum. And some regions of the nuclear membrane will be having small pores, which is called as nuclear pores. These nuclear pores helps in communicating with the other organelles that are present in the cytoplasm. Then second important organelle, it is a structure that is nucleolus. Nuclei contain relatively dense and spherical structure which is called as nucleolus. This is nucleolus. It is comparatively dense and very darker in appearance, and these are responsible for the synthesis of the ribosomes. See, it is composed of 70% protein, 70% RNA, and some small amount of DNA is present within the nucleolus, and the major function is the production and organization of the ribosomes. The genes which are responsible for the synthesis of the rRNA will be present in the nucleolus organizer region. This nucleolus organizer region codes for the rRNA that will combine with proteins and produces the ribosomes. Next, coming to chromatin fibers. This chromatin fibers is composed of protein and DNA. As I said, in case of eukaryotes, within the nucleus, the genetic material will be present in the form of chromosomes. That is, DNA and protein, they will combine together to form the chromosomes. And it constitutes about 10% of the total nuclear mass, and the average diameter of any chromatin fiber is 230 angstroms. And the arrangement or the organization of chromosome is like that. Imagine this is as an histone octamer where DNA will wound around it and form a bead-like structure. This is one bead-like structure. This is called as nucleosome. Between two nucleosomes, there will be presence of the linker DNA. So, in this way, the genetic material or the chromatin will be arranged. This looks like beads on the string. Hence, they are called as beads on string arrangement. In microscope, when we look at into the chromatin fibers, they appear as beads on string, and those beads are called as nucleosomes, and they are composed of DNA and 8 histone proteins. And in any chromatins, there is two different types of chromatin, that is heterochromatin region and also euchromatin region. The difference is that in heterochromatin is highly coiled, that is it will be very densely and highly coiled structures, whereas euchromatin, it is loosely coiled. And since it is highly coiled, it takes up more amount of stain and they appear dark during interphase, whereas euchromatin, they appear light during interphase. But in case of metaphase, heterochromatin is lightly stained and euchromatin is deeply stained. And since the heterochromatin is highly coiled structure, they allow the genes, they do not allow much of their genes for the transcription, that is they are transcriptionally very less active, whereas in case of euchromatin, they are highly active in transcription process. Next, coming to the organelle, that is lysosome. Lysosome, it acts as an intercellular digesting organelle, and it digests the organelles or any waste material by a process called as phagocytosis. Phagocytosis is nothing but engulfing. If there is any waste that is generated within the cells, it will be digested by lysosomes, and even when the cells are damaged, it will digest its own cell. And these contain hydrolytic enzymes, which is acid phosphatase, which is very important, acid DNase, acid RNase, beta galactosidase, etc. And these lysosomes are again two types. One is primary lysosomes, which are synthesized and produced by Golgi body. This primary lysosomes, they contain only the hydrolytic enzyme, whereas this primary lysosome gives rise to secondary lysosomes, which are containing both food particles as well as the enzyme. The materials that has to be digested will be present within the secondary lysosome. Then these lysosomes are also called as suicide bag of the cell, because they are responsible for engulfing their own cell if they are damaged. Next, coming to peroxisomes. Peroxisomes are the vesicle-like structure which is covered by single membrane, and they contain the enzyme catalase. These catalase enzyme, they are majorly involved in oxidizing reactions if it is taking place in the cell. And next is the spirosomes. Spirosomes are also a vesicle-like structure which is surrounded by membrane, and they are composed of about 98% of protein. The major function of the spirosome is the lipid storage. And remember that these spirosomes are present only in plants and they are absent in animals. Okay. Along with the lipid storage function, it is also having certain acid phosphatase enzyme. Next, coming to vacuoles. Vacuoles are also a membrane-bound organelle that is present in plants, animals, and some protista cells. But in plants, these are very large and they are usually single in nature, whereas in case of animals they are small and numerous vacuoles are present. Okay, inside the vacuole there is a presence of cell sap, where this cell sap is composed of sugar, salt, protein, phenols and pigments like anthocyanin. Remember, anthocyanin is a pigment that is present within the vacuole, and the membrane that is surrounding the vacuole, it is called as tonoplast. This has to be remembered. And the major function of the vacuole is maintaining high internal water pressure, and also it provides the physical support to tissues. Next, coming to centrioles. Centrioles are the hollow cylindrical structure that is present in the cell, where they always occurs in pair. That is, this can be clearly visualized during the cell division, where at opposite poles there is a paired centriole bodies, where each fibril will be perpendicular to each other. That is, one is oriented vertical to the other, and they are present only in the animal cell, and their structure is very much similar to the flagella. And new centrioles will be originating from the pre-existing centrioles, and they have 9 triplet fibrils, which each contains 3 subfibrils. This, imagine this is centriole, cross section of the centriole, where there is presence of 9 triplets. And these 9 triplets will be connected to one another, and they are also having some cross fibrils, which will give a wheel of bullock cart appearance. When we go for transverse ultrastructure of the centriole, it will resemble wheel of the bullock cart. And the main function of the centriole is the spindle formation during cell division, movement of chromosomes, and also orientation of the chromosomes during metaphase. The centriole will synthesize the spindle fibers. This spindle fibers will attach to the chromosomes, and it helps in the arrangement of the chromosomes or the metaphasic plate. And during the anaphase stage, whenever the chromosomes move towards the opposite pole, it will be guided by the spindle fibers itself. Next, coming to cytoskeleton. Cytoskeleton, it is the network of protein fibers extending across the cell and around the cell. It consists of 2 types. One, it is microtubules and microfilaments. For the first time, the term microtubule was coined by Slutterbeek in 1963. And the major function of the cytoskeleton is that it provides the structural framework and rigidity to the cell. And as I mentioned, the centrioles are present only in animal cell. They are absent in plant cell. So the function of the centrioles will be carried out by the microtubules, that is, it helps in movement of the chromosomes during the cell division. Along with that, it is also involved in intracellular transport of materials, that is, movement of biomolecules from one organelle to another organelle. For example, the proteins which are synthesized by the ribosomes has to be distributed to various organelles, that will be mediated by these microtubules or microfilaments. It is also involved in cellular motility and formation of the flagella, cilia and centrioles. These are the major different cytoplasmic inclusions that are embedded within the cytoplasm. Some are present in animals and some may be absent in plant cells. Some organelles like ribosomes, these are common to both plants and animal cells. That has to be remembered. And this is the different topics or the concepts that comes under cell ultrastructure, which is covered in both part 1 and part 2. Till now, if you have any doubt regarding any concept of the cell ultrastructure, please feel free to comment in the comment section. I'll be answering all your doubts. At last, I would like to inform about Vijeta batch that is offered by AgriAddict, where it is exclusively meant for IBPS Agriculture Field Officer exam of 2024, under which 50 plus standard mock tests and previous year question papers along with the solutions will be provided, which are prepared by top rankers of ICAR PG exam, and one-on-one doubts clearing sessions will also be conducted. So next video, I'll be coming up with the recent agricultural schemes that are benefiting the farmers in various different ways. Until then, wait for the next video. Thank you.